To produce spoken language, an organism requires a highly specific biological capability, auditory vocal learning. This is the mechanical ability to acquire new sounds from the environment and dynamically modify vocal cord output based on that auditory experience. Because human language is so complex, we might assume the mechanical prerequisites for it are shared with our closest evolutionary relatives. However, non-human primates lack the neural architecture necessary for vocal imitation. This complex capability is instead found in specific avian orders, like parrots. Mammals and birds diverged 300 million years ago, long before complex vocalization existed in either branch. Finding this capability at such disconnected ends of the tree means evolutionary pressures drove distant lineages to evolve astonishingly similar behavioral, neural, and genetic solutions for vocal imitation. Biologists refer to this phenomenon as convergent syntax. Nature faced the same computational problem twice and arrived at the same biological machine to solve it. Complex cognition and language acquisition appear as repeatable engineering solutions that can be systematically mapped across divergent species. Both human speech and birdsong acquisition follow a shared two-phase developmental model. The first is the memorization phase. Long before they make a sound, the subject passively stores an auditory memory of an adult tutor, establishing a neurological template of what the vocalization should eventually sound like. Next is the sensory motor phase. The subject begins actively vocalizing. In human infants, this manifests as babbling. In juvenile songbirds, it is categorized as subsong. These initial sounds are chaotic. To achieve clarity, the subject undergoes instruction-based learning, where early vocal output is continuously and physically adjusted over time. The subject constantly listens to their own raw motor output, compares it against the stored memory template, and refines their muscle movements to match the model. This indicates that the fundamental behavioral algorithm for learning how to speak and learning how to sing is identical. Mapping the mammalian neocortex against the avian pallium reveals how this algorithm is implemented within the brain. In humans, the perception and memory of speech are concentrated in Wernicke cilia. In the avian brain, the primary auditory processing center is the field L complex. Within this avian complex lies the NCM, storing the auditory tutor memory. The physical production of sound relies on different hardware. Human speech motor representation is housed in Broca's area, while the avian equivalent is the posterior pathway, specifically the HVC and RA nodes. This architecture creates a strict neural dissociation between perception and production. The hardware used to memorize a sound is isolated from the hardware used to produce it. This chart illustrates selectively destroying a zebrafinch's NCM. The tutor song preference score plummets. The bird cannot recognize the memory template. Yet the physical ability to sing remains intact. Pre- and post-lesion sound spectrograms show an identical acoustic structure. Erasing the memory has zero impact on motor execution. The HVC and RA motor pathway functions independently, retaining the procedural muscle memory needed to sing. In both species, the information regarding what to say and the machinery required to say it are housed in decoupled neurological systems. If memory and motor functions are physically decoupled, the brain requires a way to detect a vocal mistake and instruct the motor pathway to fix it. The bridge between these systems is the anterior forebrain pathway, or AFP. This loop, consisting of area X, the DLM, and the LMAN, connects the memory centers to the motor pathway at the RA node. Altering auditory feedback reveals the AFP's instructional role. The circuit calculates the difference between the memory template and motor performance, generating an error signal. This pulse travels through the loop to the LMAN node, feeding corrective instructions back into the motor pathway. Transiently deactivating the LMAN node immediately reverses the acoustic adjustments the bird was trying. The motor pathway loses its source of real-time correction. Human speech adjustment relies on an identical feedback architecture. When human speech is disrupted, the anterior cingulate cortex evaluates basal ganglia inputs to monitor errors and adjust vocal output. Vocal mastery depends on this real-time error correction circuit. Corticobasal loops provide the necessary mechanism for the continuous refinement of sequence generation. Understanding what governs these synaptic corrections requires looking at the molecular level. In humans, developmental verbal dyspraxia is consistently linked to a specific gene, the transcription factor FOXP2. This mutation reduces functional FOXP2 protein, disrupting the ability to execute the precise, synchronized oral-facial muscle movements required for speech. In juvenile songbirds, FOXP2 expression spikes heavily inside area X during the sensory motor learning phase. To test this genetic link, researchers used a lentivirus vector to inject RNA interference directly into area X, artificially knocking down FOXP2 levels during development. The resulting behavior mirrors human dyspraxia. The birds exhibit incomplete, variable song renditions. At the cellular level, FOXP2 acts as a transcription factor, dictating how neurons integrate signals. This directly regulates the density of synaptic spines on the neuron. The gene governs auditory-vocal integration, molding the architecture of the neuron as it learns a sound. Without this specific genetic regulation, the corticobasal neural network cannot structurally alter itself to learn speech. This multi-layered mechanical stack is shared across both species. Humans and songbirds rely on the same behavioral progression, moving from passive memorization to active sensory motor adjustment. Both rely on analogous corticobasal ganglia loops to calculate real-time errors between what they hear and what they produce. And both utilize identical transcription factors to mold the synaptic plasticity required for learning. Complex behavioral outputs, like vocal syntax and language, appear as repeatable evolutionary outcomes rather than single-origin miracles. Auditory vocal learning functions as a deterministic engineering solution, resulting from the shared constraints of physics and biology. When pressured to produce a system capable of real-time, error-corrected sequence generation, nature builds the same biological machine, regardless of the species housing it.